Well, thank you for that kind introduction. It's great to be here. I also want to, on behalf of the Smithsonian, thank Jonathan and Nancy Lee for making the connection. It allows us to be here. And I would also like to recognize that being in Kansas City is just part of what the Smithsonian is trying to do to reach out to the rest of the country and share with you some of the excitement that we have at the Smithsonian. For me personally, it's nice to be here. I have family in this area, and I don't get to see them very often, so this gives me the opportunity to be here. You also know that the Smithsonian has lots of objects. We have something over 317 million things in various cabinets and collections at the Smithsonian. But in addition to the museum function, which probably all of you understand, we also have a lot of research that goes on at the Smithsonian. It occurs in the museums, but also we have research institutes that are not located in Washington, and we do a whole range of things, and I happen to work at one of those located near Annapolis, Maryland. It's called the Smithsonian Environmental Research Center, as you see behind you, and we're a group of ecologists, and we do a wide range of things, both in the Chesapeake Bay, but all over the rest of the country, and many of us have projects around the world. So what I'd like to do is share with you some of the interest that I've developed over the years in native orchids. And I'm going to start out with a bit of an overview of what orchids are, so that that sort of brings you up to snuff, so to speak, about the biology of orchids. And then I'm going to tell you about native orchids. And there are some things about them that make them even more interesting, I think, than a lot of those that you're used to buying at the local grocery store, those kind of things. And I'm going to lead then into why I think it's necessary to have something at the continental scale to assure that our children and our grandchildren and their children are going to be able to enjoy native orchids. There are a lot of threats to them that are out there. Some of them are direct and some of them are indirect, but there's a real need to do something about that. And the Smithsonian is a wonderful platform to try to organize something that is at that scale, because basically everybody recognizes the Smithsonian, so it's a good way to get your foot in the door and tell your story to people. So with that, let me begin. We're going to talk about this family, the Orchidaceae. And there are at least 25,000 species of orchids in the world. That's probably a small number. And they represent somewhere between 8 and 10% of all the plants on Earth that produce flowers. So it's the largest flowering plant family on Earth. Some people that study grasses and sunflowers and things like that would argue with you, but I think the orchid family it's the most species-rich family on Earth. They occur everywhere. Most of them are in the tropics, where in habitats where there's not any winter, where they don't have to deal with frost. And the other thing is that many of them, at least 50% of them, are plants that occur on something else. The majority of them grow on trees and shrubs and those sorts of things. And these are the kind of orchids that, by and large, you see when you buy an orchid to take home. These are orchids that came from things that are epiphytic. They occur from sea level up to about 15,000 feet, 5,000 meters, and they occur in all kinds of habitats except oceans and true deserts. Obviously, if you don't have any water, it's tough making a living, and if you have to deal with salt, that also makes life quite hard if you're a plant, although some have figured out how to do that. I'm going to spend some time telling you about the life cycle of orchids, because that to me is the interesting part. And what makes it interesting is that orchids have a wide range of interactions with other plants, with animals, but they also have an incredibly interesting and poorly known set of interactions with fungi, and I will spend some time telling you about that. Another reason why native orchids and orchids in general are worthy of our effort is that it's one group of plants that whenever the environment starts to change, whether it's something that we do directly or indirectly, orchids tend to be the first group of plants that don't do well under these changing conditions, and they disappear. So the fact that they're very sensitive, those of us who are of my age and older in this room understand what the canary in the coal mine means. Basically, it's a barometer of change. And when the canary's in trouble, things are not going well with the air around the canary, and orchids are the same thing. When they are starting to not do well in their native habitats, that's an indication that things just simply are not going well. And in most cases, we don't understand what those problems are. It's really not a matter of people going out and digging them up. That's a bit of a problem, but there are other reasons for it that are yet to be understood. They're important. They're important from a horticultural perspective. We saw some of that today at David's place. They're interesting in terms of pharmaceuticals, the fragrance industry. Those of you who like vanilla in your food, vanilla is a vine, but it also happens to be an orchid. If you've traveled to the Mediterranean, there are places where you can have an ice cream called salop, which is made out of tubers of an orchid. So there are lots of uses for orchids. They're charismatic. We at the Smithsonian, we have the panda, and we think that's charismatic. Well, I tend to think that orchids are charismatic too, and I hope to convince you of that. They're a prominent feature of plant conservation, and the reason for that is that there are rules that regulate the international trade of all orchids. They're listed on something called CITES. So again, that's a level of recognition that this is an important group of plants in terms of human endeavors. They're in our popular culture. This orchid that you have the picture of here on the left, the ghost orchid, occurs in Florida. You'll see something about that a little bit later. But that orchid, because it is threatened and everybody was ripping it off of the trees where it occurs in Florida, there was a book written about it, The Orchid Thief, and there was a movie that was produced from the book. So it's even in our popular culture. Here's an interesting orchid. This is called Darwin's orchid. This is an epiphyte. And this was first found on the island of Madagascar back in the late 1700s. Darwin saw it and when he was on his voyage around the world. And Darwin actually wrote books on orchids. And so he saw this orchid and he said, this is a very large orchid, by the way. It has a flower about that size. And he said that this orchid has to be pollinated by an animal that has a very long tongue. And the reason he said that is that this orchid has what's called a spur on it, and that spur is about 35 centimeters long. And so he actually tried to pollinate this plant. He wasn't successful, and then he went off and did Darwin thing. And about 30 years later, they actually found the moth that has the tongue that works its way down to the bottom of that spur to get nectar. And if you go onto YouTube, you can Google this, and you will find a video that shows this happening. It happens at night, so you can actually see that. Here's one of our native orchids. You have it here in Missouri and in Kansas. This is called the cranefly orchid, and this is one of the first orchids that I did research on at the Smithsonian. Published a number of papers on it, but one of them was related to its pollination. And by the way, the shape of orchids and these pollination mechanisms are the things that have really driven a lot of the research that has been done on orchids. So in this case, here's a flower of the cranefly orchid, and it has a spur also. That's what this structure is. And if you look at, you probably can't see it from the back, but there's a little something here, and that something is the meniscus. It's basically how the tip of the nectar in the spur. And what happens is that in late July, this plant emerges and produces 35 to 45 flowers. They mature from the bottom to the top. And in the evening, if you're sitting out there quietly, you'll start to, if you're lucky, you'll see a moth coming through the forest, just kind of moving along. And then all of a sudden it gets close enough to one of these inflorescences and it does a detour, and it goes over there and it sticks its little tongue in this flower and drinks the nectar. And we did studies on this, and it turns out that during the season when these flowers are open, the moths that come to the inflorescence, they actually start exploring here first because these are the first flowers to mature, and that's where most of the nectar is. And after they've removed all of it, or these flowers have been pollinated, they don't make any more nectar, and then about halfway through the flowering period, the moths are here, and then they're up here, and then finally they get to the top of the flowers, and the flowers are smaller and they have less nectar, and the moths say, I'm out of here, and so they go off and they find some other plant to pollinate. Not all orchids offer nectar rewards to their pollinators. There's a group of orchids in Europe that are in this group called the bee orchids, the genus Ophrys. And what they do is they lure animals to them to pollinate them, but they don't give the animal anything. And so the evolution of this flower shape is that the flower looks like a female bee. Here are the antennae on the bee. And so a young naive bombus will come out of the ground and it's looking for something, and it says, I want to go to that, because the orchid also gives off a fragrance. And it lures the animal to come over to it, and the animal comes over and grabs onto this, and when it does that, it sticks its head right in there, and when it does that, it picks up pollen from the orchid, and then it'll go off to another flower, do the same thing, and deposit the pollen, and therefore you get sexual reproduction occurring in the orchid. Again, you can find videos of this on YouTube. It's quite nice to see these very naive bees trying to do something with these flowers. This is one of our native orchids. This is a Cypripedium, a yellow lady slipper. This is also an orchid that offers what looks to be a wonderful place for an animal to visit, but in fact it offers nothing. So this orchid doesn't even give off a scent. It just says, I am beautiful, come over and visit me. And so what happens is that these bees, again, they're mostly bombuses, they come over and they enter this sac. And why wouldn't you do that? Doesn't that look like a nice place to visit? So they go in the sac, and it turns out inside the sac, the labellum, are hairs that point down. And so the bee, when it gets in there, it probably figures out, well, this is not where I want to be. But it can't get out because the hairs are pointed downward. And so in order to get out, the bee works its way to the back of the flower, and right over here there's a little opening on either side. And so the bee pushes its way out. And the bee's big. And so when it does that, it touches the reproductive part of the orchid, picks up the pollen, and it flies away very angry. But it's still a young, naive bee, and so it might be attracted to a second plant, does the same thing, and deposits the pollen. So it's sexual deceit is what it is. Now this is even weirder. So some of you may know about orchid flowers and the sepals and the petals and the different parts of the flowers, but if I were to ask you what are the flower parts on this orchid, my guess is that few of you in the audience would really know what to do with that. This is a tiny little orchid about so tall that lives in Australia. In Western Australia primarily, there are seven species of this orchid, and each one of those species is pollinated by a single species of wasp, a tin-tinted wasp. And so what happens on this case is that this orchid actually has all the flower parts of any orchid flower. You see them here. But on the labellum, this is designed to look like a female wasp. And so the male wasp, and it also gives off a pheromone, a sex attractant to the wasp. So the wasp comes over, grabs onto the labellum, and tries to fly away because they mate in the air. And when it does that, there's a little hinge right here. And so what happens is the wasp flies into that, is where the reproductive parts are. And it sticks the male pollen on the back of the wasp. And again, if it decides to try it again somewhere else, then you know what happens? It produces seeds. So there are all these incredible mechanisms that have evolved with orchid pollination and orchid reproduction. But now I want to switch to the next sort of mini theme here, and to show you that even though a lot of these things have been studied in tropical orchids in particular, that we have a beautiful range of orchids in North America. So this is just a collection of pictures of native orchids. Many of these you would find them in this area. In fact, this is one that's an endangered orchid out here in the Midwest. So a little bit about orchids in the US and Canada. This is a map that shows a range in the number of species that occur in each state and province. And those of you in the back probably can't see this, but it goes from 1 to 19 at the lowest level, and more than 120 species of orchids with the highest level. And obviously Florida is the place where there are more orchids than anywhere else, because Florida, southern Florida in particular, doesn't have winter, and it doesn't freeze, and so epiphytes from the tropics can survive in Florida. But the point of this map for you is that you notice that orchids are everywhere. They're in every state, they're in every province, and the number is not small. So here in Missouri, you have about 35 species of native orchids, and about the same number in Kansas. So the point is, orchids occur everywhere. The second point to make is that of the roughly 250 taxa that we have, based on information that's available for every state and every province, that somewhere almost 60% of the native species have been identified as being in trouble. They're threatened or endangered in some part of where they occur. And there's some that are threatened, throughout their entire range. So this, from an ecological perspective, this is telling me that we ought to be concerned about this when more than half of the species in any group of plants or any group of animals are in trouble. So that's one point to make for you. And then the next map is just of the US, but this is sort of taking that information from the previous slide and asking, well, what percent of the orchids that occur in a given state are listed as being threatened or endangered and deserve being watched? And these are percentages, so the color coding is based on percentages, and you can see again that in every state in the US, some percentage of the orchids that are there are worth watching because there are problems with them. What I've done in the next 3 slides is to show you the 3 orchids that are listed as being threatened in Missouri. So the first one is the small world pogonia, and I'm going to tell you more about this orchid later on. It's one that we are doing research on. It was last seen in your fair state in 1897. Still might be here. It might be alive, and I'll give you a reason for that later on. This is the eastern prairie fringed orchid, Platanthera leucophea. It was last seen in Missouri in 1951. So it hasn't been seen for a while. And then here's the western prairie fringed orchid that people have seen. It occurs at 3 sites in Missouri. So of the 35 species or so that are in this state, 3 are definitely either gone or are in trouble. Now I'm going to switch, and I'm going to tell you sort of what turns me on in the ecology of orchids, and that is this interaction between the orchids and fungi. So orchids are like any other plant. They have seeds. The seeds germinate and they become seedlings, little baby plants. The seedlings grow and they get bigger, and then at some point they grow big enough to produce a flower, and the flowers then produce fruits, which produce the seeds, and the cycle repeats itself. But orchids are not unique, but they're quite different than just about any other plant on Earth in that they have 2 life history stages, one that is termed a protocorm, and the other is a dormant phase, and I'm going to talk about both of these. The protocorm is a particularly interesting stage because it is a stage that, following the germination of a seed, the baby orchid doesn't have any shape to it. It looks like a little round ball in most cases. But the key thing is that this protocorm stage does not have any chlorophyll in it, at least in our native temperate orchids. Now, if you don't know anything about botany, you probably know what photosynthesis is because that's what all the plants on Earth do, and that's how we get our food and all that stuff. So the only way a plant can grow up and become a bigger plant is to make its own food by capturing sunlight and then putting some water in there, and away you go. But it turns out that orchids cannot get from here to here unless they have an interaction with a fungus. And I'll talk about this dormant phase in a minute also. So we're going to put fungi into this picture, and I'm going to put now several arrows in here to show where in the life cycle of an orchid fungi are important. And you see where this is going. It's basically everywhere. Oh, I'll go back to this. The width of the arrow indicates the relative importance of the fungus in a given life history stage of an orchid. So how do we do anything with these orchids and the fungi that they have to have? It's been known since the time of Darwin that these fungal interactions occur, but it's only in very recent times, like in the last several years, that we can begin to explore the ecology of these interactions, because we have now molecular techniques that allow us to do things that we could never do before. So the way we work with this is that we isolate the fungus from typically the root of the orchid. So most fungi control the situation. They determine where on their body and when and how much and for how long they will allow this fungus to enter into their body. Typically it's in the root, and inside the cells of the root, whenever the fungus hyphae gets in there, it forms bodies that look like this over here on the right. They're called pelotons, and they're like little cottony balls. So the cells inside the orchid root become filled up with these little cottony balls, these little pelotons, which are alive. And then at some point in time, the orchid eats them. So that's how it gets its resources. It digests the orchids, or the fungus that is in their root cells. Now the way we work with this in the lab is we go out to nature and we collect an orchid root. We bring it back to the lab, and under a microscope with a little bit of training, we can train undergraduates very quickly to identify where the infection is, where the fungus is on the root. We use a little drawn-out glass slide, and we tease the little cotton balls out, and we wash them on a slide with distilled water, and then eventually, after we've done that with a few drops of water, we put the fungus on an agar plate, and you can use different kinds of media in these plates. And we do this, and if we're lucky, the fungus will grow, and it'll be clean. We won't have any contamination or anything. So we have it in the lab, but the problem is that most of these fungi do not produce things that you would call a fungus, which would be a mushroom. They don't grow up to be mushrooms, unfortunately. So when you see these things in nature, they tend to be like a blob of jelly, clear jelly on a twig or something like that in the forest. So they're very nondescript, and people don't go out searching for them, and subsequently we don't know a whole lot about them. So the only way we can identify what the fungi are is to grow them, to either take them off of the agar plate or grow them into on liquid, and then eventually we can put them in a machine that extracts the DNA of the fungus, and we get that information from the machine, and it looks something like this. You've got the little computer there, and then we send that information off to the cloud. We just say, okay, go out there, tell us what this fungus is, and then out in the cloud are data, information that people have put into that database on the DNA sequence of the fungi that have been identified. And so then the cloud sends us back a message and says, here's what you have. And so far, we have the largest collection of these fungi in my lab in the US, and 99.9%, maybe higher, of the information that comes back is new to science. The fungi have never been described. So basically everything we get out of our native orchids is new to us. We simply don't know what it is. But fortunately, we kind of learn what type of fungus is. So we get some information from that. We just don't know what the species is. So now I'm going to tell you about some of these early life history stages that we work on our native orchids at our Smithsonian site. So here are the orchids, the tway blade, which flowers in the spring, the rattlesnake orchid, which has leaves that persist all year long, the cranefly orchid, which I've already mentioned, that has a leaf in the winter. You think of that one. Why would a plant have a leaf in the winter and not in the summer? And it only produces one leaf per year per plant. Turns out if you're living down on the forest floor, in the summer, that's not a good place to be, is it? Because there's not much light when the trees produce all their leaves. So these orchids have figured out how to get everything they need in the months of the year when there are no leaves on the trees. They can make their photosynthesis. And there's another one, which someone in the audience brought up before I started. The putty root orchid is the same kind. It just has a leaf in the winter and flowers in the summer months. And then the showy orchid, which is a beautiful orchid, it comes up in the spring. It's just finishing its flowering back in Maryland. And then over here is a kind of a cute little orchid. This is called a coral root orchid. There's a whole series of, in this genus of species, the genus Corallariza. And what's interesting about this orchid is that you notice it doesn't have any leaves. And this thing is only about this tall. And what it does is it sleeps in the soil for 10 months out of the year. And then about the time the trees start getting ready to shed their leaves, this little orchid pops up above the ground, and it produces these little teeny flowers that don't get pollinated. It's self-pollination, and they produce thousands of seeds. And so this is a very interesting one. So here's a protocorm, that stage I was talking about of the tway blade. Over here you see one of the seeds, and there's an embryo in the seed. So after the seed germinates and the protocorm gets the proper fungus, and the fungus gets in through these little root hairs, they're really not roots, they're called rhizoids, and so it grows up, and then it becomes a seedling after that. We can germinate these things in the lab, and we have the fungus isolated. I'm going to tell you about that in a couple of minutes. This is the downy rattlesnake orchid, and we can grow this in the lab. Here's a protocorm, and here's a seedling. So here's the protocorm over here, and here's the first seedling stage. So we can do a little bit of this in the lab. This is this one that flowers in the autumn. It's really an interesting one. So over here in the lower left is what that protocorm looks like, and it's called the coral root orchid. So the structure of this underground part looks like a coral might in a marine environment. And so what happens in the autumn is one of the tips of this underground structure will start to swell. And it gets bigger, 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 and then what it does is it sends up this little flowering spike. And it does its thing and produces seed. But uniquely and interestingly, what happens at the end of the flowering period when the seeds are getting ready to be shed, this part on almost all the plants that we've ever looked at becomes this over on the right-hand side. It basically withers and turns brown. But before it does that, it produces these little balls that can persist in the soil for at least 3 years, completely uncovered. They're white, just waiting on the right little fungus to come by so they can grow into one of these structures. So this is sort of another mechanism for perpetuating. But what's even more important and interesting about this orchid is that it's actually a parasite on the trees. And so what happens is the trees, almost every plant on Earth has some interaction with fungi. Usually the interactions with fungi they benefit both parties. It's called mutualism. So the tree gets something, the fungus gets something. And the way the story goes is that usually the fungus gets carbon from the tree, and the tree gets water and nutrients from the fungus. And the benefit to the tree, other than getting those resources, is that the tree roots only extend so far out from the trunk of the tree. But by connecting the tree roots to fungi, what you end up doing is expanding the volume of soil that the tree can get resources from through the fungus. But in this particular case, what happens is this very smart little orchid has figured out how to sit out here on the fungus that is attached to the tree root and get everything it needs to do to complete its entire life cycle. So what it is a parasite on the tree. And the way we know that is we can do chemical analysis of the tree roots, of the fungus, and of the orchid, and they are identical. So we know that this is a parasitic kind of situation, controlled completely by the orchid. And I think it's kind of interesting that these plants can take advantage of other plants, and the other plants don't even know it's happening. So here's a protocorm of a plectrum. Here's the seed. Here's the first protocorm, or the protocorm, and here are the little rhizoids that the infection occurs in. I'll tell you a little bit about the ecology and some of the mysteries that we encounter when we're studying these native orchids. So this is a result of several years of studying this putty root orchid. And what I'm showing you here is from 2006 to 2010, the size of the leaf that the plant produced. So remember, this produces only one leaf per plant per year. And so what I plotted over here on this axis is a measure of the size of the leaf, the length versus the width of the leaf. And the bars are as follows. The black bars are the sizes of the leaves of plants that flowered in any given year. The white bars are the sizes of the leaves of the plants that were not flowering in those years. And so these are averages and some measure of how variable it was. So you can see that there's a little variation over time, and in some years those that don't flower have bigger leaves than plants that do flower, and you can explain this sort of thing. But what's most interesting is that in the years that we've been measuring these plants in the field, we've never seen a baby. We've never seen a little teeny aplectrum with a little teeny leaf, or we've never even seen an aplectrum with a medium-sized leaf, because this bar over here on the right is the average size of a plant the first time we've seen it. And you can see that the first time we've seen it, no matter what year it occurs in, it's as big as everybody else who's been there for however long they've been there. We have no idea what's going on below ground. How a plant this orchid can be below ground and all of a sudden emerge and be as big as every other orchid in that population. We think it has something to do with the fungus, but we haven't worked that all out yet. Here's the cranefly orchid, the tipularia. I've told you a little bit about this. Here's the protocorm, here's the first root, and here is the first leaf. And you can see the size over here. This is about 1 centimeter. For years, we never saw any babies of this orchid also, but when Hanna Rasmussen came over from Denmark to work with me, we were walking in the forest one day, and here I am at one of the tree falls in our forest, and what I'm doing is I'm pointing at a baby tipularia. And once we figured that out and got the search image for where little orchids were, we found out that almost exclusively they occur on decomposing wood, twigs and logs and things like that. And so now that we know that, we can find them readily in nature. But what's most interesting is that when we get the fungus out of the baby tipularias and do its molecular analysis and send it off to the cloud, the information that comes back, the cloud can't even tell us what group of orchids it's related to. So this is something that's completely unknown to science. Now this is kind of complex. Don't get worried about it. What this is a genealogy. And so these lines show how related one thing is to something else. And down here on the lower left, what I've got in this color are the fungi that we have isolated and sent off to the cloud for this coralloid orchid, the Corallorhiza. And what you can see over here is that this group is not very closely related to anything over there. This is the parasite, and it's using a group of fungi in this genus Tomentella. So that's the first point to make. Over here in the red these are fungi that we've isolated from the tipularia, the cranefly orchid. The green are fungi that we've isolated from the tway blade, and this color, this sort of yellowish, is from Goodyeara. So the 3 orchids that I've been telling you about. Now the way you would read this is as follows. Tipularia has its own group of fungi. They're all in this related to this genus called Tulisnella. It has its own group of fungi. This group could be one species of fungus. That could be another fungus. We don't know because people haven't described these things. But basically, they're all sort of down here. This is a fungus from Georgia. This is one from Maryland over here. But you also notice that this orchid can use the same fungus that is with Liparis, and it also can use the same fungus that Goodyeara uses. So this is an orchid that can use more than one orchid to meet its needs. The Goodyeara, all of the fungi we are showing here are related, very closely related to each other, from North Carolina, from Maryland, from Georgia. But you'll notice this line has little ticks on it here. What this is telling us is that the DNA of this fungus is just a little bit different than that one, and it's just a little bit different from that one. We don't know whether these are different species. All this is telling us is that molecularly they're a little bit different from each other. The Liparis, in contrast, you look at this line and there are no little ticks on it. And this is an orchid that is very specific. It only uses one fungus. It needs that fungus at every stage in its life cycle, and it doesn't matter where you get that fungus from, it is genetically the same. So in this case, the fungus from Liparis is the same whether it's from Virginia or Iowa or Michigan or Maryland. So this is a very one-to-one situation, and if you don't have the fungus, you clearly won't have this particular orchid growing in your neighborhood. Now I'm going to tell you a little bit about one of our ongoing projects with this orchid that I showed you as being still on the list of threatened and probably extinct species in Missouri. It's the small world Pogonia. This is listed federally. So this was listed by Uncle Sam as an orchid that was initially, when it was listed as almost on the verge of extinction. So, all the bells and whistles went off. And then somebody up in Maine, they did a study where they went to places where the orchids occurred, and they built a model. And then they used that model to predict other places in the environment where you should find the orchid because it only occurs on north slopes and slopes of a certain, whatever the environmental information was. And they went out and they looked for the orchid, and they found it in more places. And so what happened was the federal government lowered the ranking of this particular orchid from endangered to threatened. So it's currently listed as being threatened. And there are not many orchids that are listed at the federal level. That process is more of a political process than anything else. But this is basically an orchid that's in trouble. And we've been working at it with a grant from the National Park Service, and we also work with the Army at Fort A.P. Hill near Richmond, Virginia. So this is the original range of Isotria. You can see it's out in your part of the world. But what's interesting is it occurs from the south, Georgia, all the way up to Maine. And this is the only plant that I'm aware of when they talk about what a viable population is. The definition for a viable population in the south is different than the north. To be a viable population in the north, you have to have at least 40 plants in a population. And in the south, it's 20 plants. Now, how they came up with that, I have no idea. Mostly because they didn't find many plants in the south, and they ended up doing that. So we're studying this orchid in populations at these two sites. And over here on the left are data for the populations at Prince William Forest Park, 4 different populations. And the 3 on the right are from Fort A.P. Hill. And the numbers with each of the bars are the total number of plants that we've found. And you can see the numbers aren't very big. The largest population we started working with had 37 plants in it. And the yellow bars tell you how many plants in a population have flowered. And if you look at the scale over here, you see it's a much smaller number. So what this is telling us is that even though the populations are small, very few of them are producing flowers. And the green bars are how many of them have produced fruits. And you can see they're almost producing no fruits, just 1 fruit maybe in a population a year. So this plant it doesn't reproduce any other way. It's only by seeds, and it's not happening at a very high level in the southern part of its distribution. So here's what we know at this point in time, is that many of these plants appear above ground for only 1 or 2 years, and that goes to what I was telling you earlier about this dormant stage that a lot of our native orchids have. So when we find an orchid, the probability is very high that we're only going to see it for 1 or 2 years, and then it's going to disappear. And we map these things so we know where they are. And over the 6 years or so we've been doing this study, no plant that has gone dormant has ever appeared above ground. And it's possible that they could be living below ground, and we have indirect information that that's very likely. One is that when we go to these populations, almost every year we find a new plant that is growing next to the ones we've been looking at, and we never saw that plant before, and so it's been living below ground for at least 6 years without producing a green leaf. And then I have a colleague at the University of Maryland who studies the pink lady slipper in Virginia, and he has mapped plants for the last 40 years almost, and he has some plants that he mapped them initially, and they disappeared, and they came back 25 years later, which is kind of incredible. So the message I want to leave with you there is this fungal thing is really important to our native orchids. Now, for this particular orchid, it's important for us to know what fungus it associates with if we want to have any chance of conserving it or restoring it. And so we kind of know that now. We know that it's in this group of fungi called the Russulaceae, and this is a russula over here in the picture. But the sad part of this is that all of the fungi in this group are the ones that form these connections with roots of trees and shrubs. And they're called ectomycorrhizal. That's a mouthful. And, but the thing is, we have not yet figured out how to grow those fungi in the lab. So we can't do experiments with this because we can't grow the fungus yet. But even more confusing is we had a master's student from the University of Connecticut work with us the last couple of years. She's just finished her thesis. And it turns out that it would have been nice if all we have to know is that the fungus is present or it's not present. If it's present, the orchid has a chance. We have evidence from the isotry and one other orchid that it also may be the quantity of fungi that is present that's tied into whether the orchid is going to make it or how well it's going to do. And here's an example of that from work in New Hampshire and our work in Virginia. So the y-axis over here is how much fungus is there. So we have molecular tools now. We can just take a soil sample and extract the DNA, and we can determine how much fungus is in there, not just whether it's present or not. So the tall bars in both cases are how much fungus is present next to the plant that was green that year. The second bar from the right in is how much fungus was present this year for a plant that was present last year but did not produce a green shoot this year. And you can see it's always less. And so basically the longer the plant has been underground, as you go from right to left, the less fungus is there. So we need to know much more about what's going on with these fungi, how to encourage them to grow and persist in these environments. So to sort of summarize this little part, we know that the plants are doing much better in the north than they are in the south. More plants in New Hampshire and Maine produce flowers. More plants in Maine and New Hampshire produce seedlings that people have found. And fewer of the plants in the north are entering this dormant phase. So what this tells us, which I think we already knew, is that in the south, these plants are in trouble. But that's not the end of the story. I had a former postdoc, Makihiko Ikegame, a Japanese fellow. He has the skills where he can take information on weather. And he can look at places where people have found orchids and find out what the weather conditions are at that place, put that into a model, and then he can produce a map like this, which is the distribution of the orchid based on weather data. And it turns out this distribution very closely matches the actual distribution of this small world Pogonia. So his models do a very good job. And I asked him to project out. You know, we're in a world where the climate's changing a bit. And so what's it going to be like for this plant in a few years? And this is 100 years from now using one climate model. And you see there are almost no plants left in the south. It's migrating north, and it's migrating to the northwest, Wisconsin, Michigan, Minnesota. And think of that for a minute. How is the little old plant from Fort A.P. Hill in Virginia going to migrate to Minot, Minnesota? It's going to be a tough journey because there are a lot of cities, a lot of pavement, a lot of cornfields, soybean fields, everything's in there. So for our native plants to make it in the future, in whatever that climate is going to be, I don't think it's going to be a simple story. We're going to have to, as humans, get involved in that process. So to summarize the part on this isotry, what I'm going to do now is tell you, after working on this species for 6 years diligently, not only us but other people, I'm going to show you which of the connections we know something about. So in Virginia, that's how much we know. So I don't think this is unusual. So here's the overall on this is that I think our native orchids, understanding their ecology from a conservation perspective, it's like a jigsaw puzzle. And in order to enable these plants to survive, we have to put the jigsaw puzzle together, and we haven't done it for any species. We're not there yet. And there are very few people doing this kind of research. So the climate's changing, we're changing the world. How are we going to stay in front of that so that we can assure that these orchids are going to survive? I used to use some sleep over this. I still do, but I think there's hope now. So a new program at the Smithsonian has enabled me to do something I've always wanted to do, which is to bring botanical gardens and people, the general public from all over the country, to help us figure out how to conserve native orchids. So we've formed this organization called the North American Orchid Conservation Center. It's initially started by funding from the Smithsonian and the U.S. Botanic Garden. So what I'd like to do now is jump out of this, and this is actually you should have had people keep their cell phones on, because now you could keep them in the where they won't ring. But now I want you to go to the website of the North American Orchid Conservation Center. Not really. So what I'm going to do now is jump out of this, and I'm going to show you a video. It's a 4-minute video that describes what it is we are attempting to do.
For thousands of years, orchids have captured the imagination. Their unusual flowers have inspired artists, poets, and horticulturalists across the world. The orchids of the tropics have become famous for their intricate shapes and vibrant colors. But farther north, many of the estimated 250 species of orchids in the United States and Canada are fighting for survival and risk dying out with their beauty unseen.
So knowing that over half of the species of orchids that occur in the U.S. and Canada are endangered is a red flag to us. It tells us that we really need to understand their ecology, we need to know how to conserve them, and we need to know how to restore them.
Dennis Whigham and his colleagues have spent more than 3 decades studying native orchids in the mid-Atlantic. But saving biodiversity across the whole of North America required something on a larger scale. In 2010, Whigham and other collaborators from the Smithsonian and the U.S. Botanic Garden teamed up to form a continent-wide effort to conserve native orchids. They called their network of researchers and botanic gardens the North American Orchid Conservation Center, or NAOC. Its mission, to ensure the survival of all native orchids in the U.S. and Canada.
We have a series of goals, and we're going to reach those goals by establishing partnerships that are national in scope. We will work with our partners to eventually assure the survival of all of our orchids through a whole series of activities ranging from seed bank collections to collections of fungi that interact with orchids, to actually figuring out how to grow and sustain orchids so that they can be used for education purposes as well as for restoration purposes.
For the North American Orchid Conservation Center, ensuring the survival of all orchid species is no simple task, especially with the rich diversity of species they're trying to protect. There are many native orchids in peril. Some, like the dragon's mouth orchid, have already disappeared from Maryland. In Florida, they are struggling to protect the ghost orchid, a leafless orchid that haunts the Everglades. It is found nowhere else in North America. Another challenge for conservationists, orchids, perhaps more than any other plants in the world, have a deep and complex connection to the forest, particularly when it comes to the fungi they need to survive.
One of the challenges that orchid seeds have to meet is that they're very small and they contain very little food nutrients. In order for the seeds to germinate and grow in nature, they depend on a symbiotic partner. It's actually a fungus that the orchid digests. It provides carbon and allows the seed to germinate and grow.
For NAOC to fulfill its mission, it will need support from outside. Initially, resources in the form of grants, contracts, and contributions will allow NAOC to hire a director, establish a national collection of orchid seeds and symbiotic fungi, and develop an interactive website for the public. Additional resources, including the establishment of an endowment, will assure NAOC's future success, preserving our orchid heritage.
The analogy that we like to use is that our native orchids are similar to the canary in the coal mine. It's the one group of plants that are sensitively adapted to the environment that they live in for a number of reasons, because of specialized pollinators or fungi that they require at different stages in their life cycle. But the fact is, when the ecosystem starts to be degraded, the orchids are among the first plants that begin to disappear.
The successful establishment and development of NAOC will assure the survival of the largest and most charismatic plant family on Earth.
Okay, so I encourage you to visit our website if you want to learn more about the kinds of things that I've been talking about. We also have a tab on there that has marvelous pictures that amateurs provide to us, just beautiful orchids. The other thing I want to tell you about, and another reason why you should have kept your cell phone out, is that we also have launched this website. It's called Go Orchids, and the goal of this website is to allow anyone anywhere in the U.S. and Canada to identify any orchid that they see. And this is the front page of the site, so you can go into this, and you can go in by where you are, location. So if you open this website up and go into Missouri now, up will come a picture of every orchid in Missouri. And you could enter it by name. There are other ways to go into it, so I'll quickly sort of go through this, I think. Why we're not working there. Whoop. Okay, so this is an example of where you would go in by where you're located. So you enter in a name, say Maryland. When you push in Maryland, up will come a picture of all 52 orchids that occur in Maryland. And if you recognize the one that you have right away, you just click on the picture, and up will come a species page. So, or you could go in by name. You could type in this name, which is the pink lady slipper. It'll take you to this page, and then you click on that, and it'll come up with a species page where you have these beautiful pictures of that orchid streaming in front of you. And then as you scroll down through the page, it'll tell you everything we currently know about that orchid, whether it's endangered or threatened, and whether it has, we know about its fungi, whether we know about its pollinators. Or you can play botanist, and you can go in and you can ask questions, and this is a smart website, so you can say, I have a pink flower. I'm in Missouri, I'm looking at a pink flower, so it'll take all the 35 orchids of Missouri, and then all the pictures will disappear except the 3 that are pink. And you'll say, well, that doesn't quite look like it. Well, how many leaves does it have? And you have a choice, and you say it has X leaves, and then it'll get rid of 2, and then you have 1, and then you have your orchid. And by the end of this calendar year, we will have all of the orchids in North America and Canada on this website. They currently have the orchids of the Mid-Atlantic states, New England. We just launched Alaska. I'm going to be up there in about 2 weeks working on some orchids. So this is an exciting website that we're going to be using for a variety of purposes, primarily to educate the public about orchids, but also to get people interested in botany, and so we can have a little higher level of botanical literacy in the country. So the last thing I want to do is to tell you where this organization stands. So we've just finished the first phase, which is the stuff I've been telling you about. We're entering the second stage, and what we're doing here is we're developing partnerships in regions around the country. We're developing the seed banks and the fungal banks, and this is where the rubber is going to hit the road. We're going to start getting materials flowing into us to work with botanical gardens so that they can work with us to eventually learn how to propagate all these orchids. Ecologically, where that's important is we want someone in Kansas and Missouri to grow these native orchids in their botanical gardens so that they create sustainable populations. Most botanical gardens, they put a plant in, and you look at the plant, and the plant dies, and they put another plant in. What we want is sustainable populations. We want to know about the fungi and the soil characteristics, so we want to figure out how it works. And we think where the botanical gardens will give us a great step up is that if we can figure that out in botanical gardens, when it comes time to restoring these plants in nature, we're going to learn a lot. Because as I've tried to show you with this small world pogonia, unraveling all the secrets of nature and figuring all this stuff out is very, very difficult. And we think we can get a better hand up on it by going the other way. So then we have a third phase. We want to have an endowment and just continue to do the education and outreach and growing this so that 20 years from now we can say we did it. We have all 230 orchids growing successfully someplace, and we have examples of restoration that's been successful that we can show you. A few people to thank, the funders there on the top, and the people that really made this work is our technician Jay O'Neill and Melissa McCormick, who does all the fungal work, and Hannah Rasmussen, who got us into this. And then we have other partners within the Smithsonian. This is a collaborative effort between several parts of the Smithsonian and the U.S. Botanical Garden. So that's the last slide. I hope I've been able to demonstrate to you that these are really interesting plants, that they're worthy of our attention, and that what we're trying to do at the Smithsonian is something that, from a national perspective, is really a worthy goal. And if any of you know of organizations and individuals who want to help us in this endeavor, please feel free to contact us. And I'd like to thank you all for attending, and if we have any time, I'd be happy to address questions. Thank you.
If you have a question, raise your hand. I'll come by with the microphone. Okay, I'll come back up. I lost track of where.
Thank you. Are you studying the orchids in Hawaii?
There are only 3 native species of orchids in Hawaii. A collaborator of ours at Illinois College is working on actually restoring one of those species. They've grown the plants, they're putting them back in Hawaii. They still haven't quite got the fungal side of it out, but yeah, we want to work with those 3 species also.
We have a question on your right here.
Well, Dr. Whigham, I was really impressed to learn that some orchids are in the dormant stage for 25 years, and I think for a nutrition and reproductive specialist, that's not a bad way of survival. So I was interested in the anatomy of those dormant plants, and if that's too botanical, I'm glad to take the answer off the air. But is one of the reasons why they are endangered that the fungus is the problem child, or is that something of your jigsaw puzzle you have not determined? I could imagine that the fungus, like you said, works with various species, and as fungus are destructors, maybe they feed off some other organisms and don't need the orchid necessarily.
Well, you've opened a can of worms. The answer is we don't know, but we've published papers, for example, where we've looked at what happens in bad years. So in Maryland, you know, we're hot, humid, we get a lot of rain, but we have had droughts. And so we go through that in some summers. And we've done some research where we've taken plants that we work with every year, and we've taken roots from those plants, and we've looked at whether they have a fungus in them or not. It turns out that in the fungus is the weak partner in this deal. And so whenever the environment starts to change, like a drought, what happens is the orchid it digests all the fungi, and then it goes through a period when it doesn't have any fungi. And then after the good growing conditions come again, the orchid can take up a fungus, and we've shown that in a few cases it'll take up the same fungus molecularly that it had before, but most of the time it'll pick up another fungus that is related but is a little bit different. So that tells us that the fungus is the weak member of this, and I think you're exactly right that it could be that changing conditions that are resulting in fungi not being present may have a lot to do with this. So we really need to know what the substrate features are that enable the fungus to proliferate. There's another part of this which is strange, is that I don't know how many of you are aware that most of the worms in your soil are not native worms. Most of them are from someplace else, mostly from Europe or Asia. At our place, we have about 35 species of earthworms. Only 3 are native. And we've published some results. Well, it turns out most of the fungi that the orchids use are in about the top 5 to 10 centimeters of soil. And what these non-native earthworms do is they gobble leaves. They consume the leaves in the forest floor. And so some of our forests that have the right mix of tree species for the earthworms, by the middle of summer, there's hardly any leaf litter on the forest floor. The other thing that happens is you know how an earthworm makes its living, most of them, they gobble soil. And so what they do when they're gobbling the soil is they're changing the microbial community in the soil. And soils that have a lot of earthworms, they are switching from a dominance of fungi to a dominance by bacteria. So that could be affecting this whole thing. The other thing that the earthworms do is when they're moving soil up and down in the soil profile, they passively eat these tiny little orchid seeds. And they take them far enough down into the soil where when it comes out the other end of the earthworm, it's below the level where the fungus is. So they consume the seeds, and some of them get destroyed when they're passing through the earthworm's stomach, digestive system. So that's going on in our world too. So we're doing all kinds of things, and I think the fungus side of it is really key to a lot of this. By the way, there's an orchid in Australia that spends its entire life below ground. It never comes above ground. It produces a flower below ground. It produces seeds below ground. So that really tells you that the fungus works. Yes.
I have a question about soil disturbance. You know, like if an area has been disturbed, like an old home site, and it's gone back. And one of the places I've been, they found that there's been orchids showing up there as a result, I presume, as a result of the soil being disturbed and being then allowed to return to natural setting.
Yeah, I don't know what the answer to that specifically would be, but there's sort of a bright side to the story in that these tiny orchid seeds, when they're being dispersed, which is typically in the autumn, if they're dispersed by wind, and most of them land near mummy, but some of them can get taken up in air currents, and they can go long distances, probably hundreds of miles. And so it's possible that they can move through seed dispersal. And we've actually had 2 orchids show up at our site in Maryland. One of them, the closest known population of that species is 300 miles away. And we had another orchid appear that doesn't even exist, there are no populations in the county where we are, and it just appeared in a garden. So there's hope. I think if we can keep the soil happy and healthy with the right fungi, things can happen.
Yes, ma'am.
Are there any other plants that have the relationship with fungi?
Almost every plant has relation. Probably the last estimate I saw is 85% of the flowering plants on Earth have they have fungal connections all the time, and most of them are what I referred to early on as mutualistic, that both parties benefit.
Any other questions? Well, thank you, Dr. Whigham, for a wonderful lecture.
Thank you again.
And thank you for attending tonight's event. As a reminder, I forgot to mention this during my introductions, but e-tickets for our fall lectures will be online July 1st at 9 a.m. So check our website, lindahall.org, July 1 at 9 a.m. Thank you and good night.